Imagine for a second that you, or maybe someone you love, is going through cancer treatment.
Yeah.
You walk into the clinic, you sit down to the chemo or the radiation, and, well, you naturally assume that these powerful treatments are doing one very specific job, right?
Yeah. You think of it as a search and destroy mission.
Exactly. You picture this microscopic battlefield where the enemy cancer cells are just violently destroyed, like completely wiped off the map. But what if those treatments actually leave behind this, like, army of zombie cells? Cells that don't die, but instead freeze in place, stay incredibly active, and actually start hacking into your own immune system.
Yeah, it forces us to completely rethink everything we thought we knew about oncology, honestly. We're so used to looking at cancer treatment as this binary outcome. You know, the cell is either alive and dividing or it's dead and gone. But this introduces a massive, highly dynamic gray area.
So today, we are taking a deep dive into therapy-induced senescent cancer cells, or TISCCs. These are the zombie cells. They've stopped dividing because of the therapy you received, but they are metabolically wide awake. And our mission today is to really explore the dual nature of these cells. We're going to look at how they actually start off as these powerful allies, calling in your immune system, but then over time, mutate into chronic enemies that protect the tumor.
And crucially, we'll get into the cutting-edge therapeutic strategies, what we call time-aware therapies, that scientists are developing right now to finally defeat them.
Okay, let's unpack this. I really want to start at the cellular level. How does a cancer cell even become a zombie in the first place? Like, let's say a breast cancer patient gets a standard chemotherapy drug, like doxorubicin. What exactly happens?
Well, the foundation of this whole process is something called the DNA damage response, or DDR. When a patient gets radiation or a powerful chemo drug like doxorubicin or paclitaxel, the mechanical goal of that drug is to severely damage the tumor's DNA. I mean, we are talking about causing double-strand breaks.
So the DNA helix is literally snapping in two.
Literally snapped in half, yeah. And that physical snap is immediately detected by the cell's internal sensors. There are these specific kinase enzymes, ATM and ATR, and they basically patrol the DNA. When they feel that mechanical break, they sound a massive alarm.
I always like to visualize the cancer cell as this out-of-control runaway train. The chemotherapy comes in and violently blows up the tracks. Now the train can't keep moving forward, so it has to react.
That's a great analogy. And the reaction is an emergency lockdown. That ATMAT alarm cascade activates major tumor suppressor pathways. The most famous ones are the p53, p21 pathway and the p16RB pathway. You can think of p53 as the ultimate emergency brake. It slams down and forces the cell into a permanent state of cell cycle arrest. And the biological term for that permanent arrest is senescence.
So the runaway train screeches to a halt. It's not dividing anymore. But, and this is the crazy part, the engine is still revving, the exhaust is billowing out, and the horn is blaring nonstop.
Yeah, and that engine revving isn't just noise, it's physically and chemically remodeling the whole cell. If you look at these senescent cells under a microscope, they look bloated. A big reason for that is their lysosomes, which are basically the cellular recycling centers. They just go into absolute overdrive.
Because they're trying to clean up the mess from the chemo.
Exactly. They're desperately trying to process all this internal damage, but they get overwhelmed, they expand massively, and they create this classic chemical marker called SA-beta-Gal. Scientists literally use this marker to find zombie cells in tissue samples. And because these recycling centers are so hyperactive and inflamed, the cell starts pumping out this vast, complex cocktail of proteins.
Right, which brings us to the SASP, the senescence-associated secretory phenotype. That's the billowing exhaust I was thinking of. It's just this massive cloud of chemicals.
The SASP is everything. It's how the zombie cell manipulates its surrounding environment. The cell uses this chemical cloud to broadcast its damaged state to the rest of the body. Therapy-induced senescence isn't just some passive, side effect where the cell gives up. It's an active, incredibly potent survival program.
Okay, so just to do a quick recap for you, the listener, before we move on, think of the STOP mnemonic here. S, snap the chemo, snaps the DNA. T, trigger the ATM sensors, trigger the p53 emergency brake. O, overdrive, the lysosomes swell up. And P, pump the cell, pumps out that toxic SASP cloud. It has officially become a zombie. Now, instinct might tell you a zombie cell is purely a bad thing, but initially this is actually a good thing, right?
Oh, absolutely. Immediately following treatment, the zombie state functions as this massive danger signal to the immune system. We refer to this as the immunogenic phase, or the good phase. And the mechanism behind this is just fascinating. It begins inside the zombie cell's nucleus. The protective inner wall of the nucleus, which is maintained by a protein called lamin B1, starts to literally crumble
Wait, so the cell is losing its structural integrity from the inside out?
Precisely. It loses lamin B1, the wall crumbles, and because of that, fragments of the cell's own DNA begin leaking out into the main fluid body of the cell, the cytosol. We call these cytosolic chromatin fragments, and this is an enormous red flag.
Because DNA shouldn't just be floating around in the main body of the cell.
Right. Human cells have this built-in ancient evolutionary sensor called the CGA stinging pathway. It was originally designed to detect viral infections because, viruses inject their DNA into a cell's cytosol. So when the zombie cell's own DNA leaks into that space, it accidentally trips this ancient antivirus alarm.
Oh, wow. So it essentially tricks its own internal security system into thinking it has a viral infection.
Yep. And the response is super aggressive. The tripped CJS sting alarm causes the cell to produce and release a powerful immune-activating chemical called interferon beta.
I was reading about interferon beta in the source material, and it seems to function almost like a flare gun going off in the dark. It's illuminating the tumor for the immune system, basically signaling that something is critically wrong right here.
It illuminates the area, and more importantly, it actively prepares the cell for inspection. Interferon beta forces the zombie cell to drastically upregulate these things called MHC-I and MHC-I molecules on its surface. You can think of MHC molecules as like microscopic display cases.
Display cases for what?
For the tumor's antigens. The zombie cell actually chops up its own internal mutated cancer proteins, places them inside these MHC display cases, and pushes them to its outer surface. It's physically presenting its own identifying cancer antigens to the immune system's assassins, the CD8 positive and CD4 positive T cells. It's essentially saying, here's exactly what the enemy looks like.
That is wild.
Yeah.
And the SASP chemical cloud we discussed earlier, that's working in tandem with this, right?
Yeah.
The sources mentioned it releases specific homing beacons like the chemokine CCL2 to draw in other immune cells like natural killers and macrophages.
Yeah, it's a highly coordinated, multi-layered distress signal. And on top of that, the zombie cells are also releasing these tiny packaged bubbles called SENEVs, senescence-derived extracellular vesicles. Think of them as microscopic thumb drives.
Thumb drives, okay, I love that. What's on them?
They contain specific molecular instructions, and they transfer them directly to dendritic cells, which are kind of like the immune system's intelligence officers or generals. This intel transfer helps the generals organize a perfectly targeted T-cell attack against the tumor.
Wait, if we pause here, I have to ask the obvious question. If these zombie cells are firing off flare guns, putting the cancer's exact profile in display cases, and handing over thumb drives with targeting intel to our immune generals, why isn't the cancer just cured right then and there? Why does the tumor survive this?
What's fascinating here is that it all comes down to the biology of time. The immune system is designed to respond to acute short-term emergencies. Acute inflammation clears out threats quickly. But if those zombie cells aren't cleared out rapidly by the immune system, they linger. And as time passes, the nature of their chemical exhaust changes. It turns the environment from acute inflammation into chronic inflammation.
So the flare gun runs out of ammo, and the toxic smoke screen starts.
That is a perfect way to look at it, yeah.
Let me just do a quick recap for you guys listening. In this good phase, the nuclear wall breaks, DNA leaks out, trips the CGAS sting virus alarm, and shoots the interferon beta flare gun. The cell puts cancer proteins in the MHC display cases and recruits T cells. It's an incredible defense mechanism. But as we just heard, there's a timer on it. Here's where it gets really interesting. What happens when that timer runs out?
If the zombie cells persist, we enter the immunosuppressive phase, the bad phase. The composition of that SSP chemical cloud shifts dramatically. Instead of pumping out alarms, the zombie cell begins secreting high levels of chronic inflammatory cytokines, particularly ones called IL-6 and IL-8.
And based on the research, this is where the environment turns hostile to our own body. The IL-6 and IL-8 essentially turn the tissue around the tumor into a toxic swamp. A swamp that repels the good immune cells and actually invites in bad actors.
They turn the tumor into an impenetrable fortress. Those cytokines recruit MDSCs myeloid-derived suppressor cells, which actively shut down immune responses. Even worse, they brainwash the local macrophages. Normally, macrophages act as the garbage disposals of the immune system, right? Eating up diseased cells.
Right.
But the zombie cells chemically reprogram them into what we call M2 polarized macrophages. Instead of attacking the tumor, these reprogrammed macrophages start protecting the cancer and releasing growth factors to help it survive.
It's basically a hostage situation where the first responders are brainwashed into joining the enemy. And they also build a physical barricade, don't they? The sources highlight a specific cytokine barrier they deploy.
Yes, the zombie cells secrete a molecule called CXCL12. This chemical interacts with receptors on the surface of the good T cells, specifically a receptor called CXCR4, and it effectively downregulates or strips those receptors away. Because of this chemical mismatch, the T cells lose their ability to navigate. They are physically blocked from migrating into the tumor nest. They just get stuck on the outside, wandering aimlessly.
A microscopic force field. Man, that is grim. But it gets even darker. The zombie cells also put on body armor. They massively upregulate proteins on their surface called immune checkpoints. We hear a lot about PD-L1 in cancer commercials, which basically tires out the T cells. But the zombies also use a signal called CD47.
CD47 is infamous. It's known as the "don't eat me" signal. And in zombie cells, it's actually supercharged by a specific modification called QPCT. When a macrophage approaches the zombie cell to clear it away, the CD47 protein on the zombie surface binds to a receptor on the macrophage. It acts like a molecular stop sign, paralyzing the macrophage and forcing it to leave the zombie alone.
So the zombie cell actively prevents its own execution.
Exactly.
I spent a long time trying to wrap my head around this next piece of the puzzle because it feels like straight-up biological alchemy. Let's talk about the CD73 adenosine axis.
Oh, this might be their most brilliant survival mechanism. So the zombie cells elevate the production of an enzyme called CD73 on their surface. Now, the body naturally uses a molecule called ATP as its primary energy currency, right? It's the battery power for our cells. CD73 captures that ATP floating in the surrounding environment and metabolizes it. It strips it down into a totally different molecule called adenosine.
Wait, so if ATP is energy, they're literally taking our own biological battery power and reverse engineering it into a chemical weapon.
That is precisely what is happening. In the tumor microenvironment, adenosine functions as a potent chemical sedative. It just floods the local area. So any T cells that actually manage to survive the force field and the brainwashed macrophages are suddenly bathed in adenosine, which tranquilizes them. It completely shuts down their ability to fight.
They turn the rescuer's own adrenaline into a tranquilizer dart. That is unbelievable. Just to take a breath and recap this transformation for you. The zombie cells stick around too long. Their exhaust turns toxic with IL-6 and IL-8. They brainwash the local macrophages. They deploy a CXCL12 force field to keep T cells out. They flash CD47 don't eat me signs. And they convert your own energy into a sedative to put the immune system to sleep. They have effectively insulated themselves from the cure.
And unfortunately, they don't just stay in a defensive crouch. They can actively go on the offensive.
Yeah, this is the part that completely changes the narrative. We established early on that these cells are in permanent arrest. They cannot divide. But the source material shows they can still drive metastasis, the physical spreading of the cancer to new organs. In things like thyroid cancer, these non-dividing zombies act as leader cells.
Right. Even though they are incapable of dividing, their toxic SASP cloud contains enzymes that degrade the extracellular matrix. That matrix is the fibrous scaffolding that holds our tissues together. The zombie cells literally melt a pathway through the tissue scaffolding. The active dividing cancer cells that survive the initial treatment then use that freshly carved tunnel to escape the primary tumor and invade the rest of the body.
But the wildest mechanism they use to clear that path involves weaponizing another type of white blood cell entirely, the neutrophil. The process is called mitosis.
Neutrophils are typically our biological first responders to bacterial infections, but the zombie cells use their chemical exhaust, specifically factors like CXCL8 and GCSF, to deliberately lure neutrophils deep into the tumor. Once the neutrophils arrive, the zombie cell triggers them to undergo mitosis.
Can you explain the actual mechanism of mitosis? Because reading about it, it sounds like a kamikaze mission.
It essentially is a kamikaze mission. The neutrophil violently ejects its own DNA along with a bunch of toxic proteins out of its cell body to form a sticky, fibrous net in the surrounding tissue. It's called a neutrophil extracellular trap, or NET. Now, in a normal infection, this net catches and kills bacteria.
So in this context, is that net a literal physical mesh that catches our T cells, like a microscopic barbed wire fence, or is it more of a chemical hazard?
It acts as both, which is what makes it so devastating. It's a dense physical mesh of sticky DNA that physically tangles up incoming T cells. But that mesh is also studded with highly acidic, toxic proteins that chemically suppress those T cells and further dissolve the tissue scaffolding.
So the zombie cell lures in a guard, forces it to explode into a barbed wire trap, which stops the rescuers and melts the walls of the tumor so the cancer can escape.
And that tissue damage from the nets can actually awaken other dormant cancer cells that were hiding in the tissue, leading to a massive, aggressive recurrence of the disease. It's a vicious, self-reinforcing loop where the body's own defense mechanisms are just completely weaponized.
It is a terrifyingly dynamic ecosystem. Let's recap this wildcard phase really quickly. Neutrophils get lured in, they undergo this kamikaze netosis, spitting out sticky DNA nets that block T cells, melt tissue, and awaken dormant tumors. Okay. We have this enemy that changes its strategy based on time, manipulating our own biology at every step. So what does this all mean for how we actually treat patients?
It means the old playbook has to be thrown out. Historically, when clinical trials tried to target senescent cells, they failed. And the underlying reason for that failure was treating senescence as a static, permanent endpoint. They just brought a sledgehammer, assuming a zombie cell on day 1 was the exact same thing as a zombie cell on day 50.
Right. But as we've explored, these cells evolve.
Exactly. So the solution researchers are developing now is a time-aware therapeutic framework. We have to match the weapon to the ticking clock.
You have to fight a dynamic enemy with a dynamic strategy. Let's walk through how this actually plays out. It starts during that early window, right? When the zombie cell is still acting as a flare gun and displaying the cancer's profile to the immune system.
Right. During that early immunogenic window, the goal is to capitalize on the alarm. You do not want to kill the zombie cell yet. Instead, you administer immune checkpoint inhibitors or specialized senescent cell-based vaccines right when the cell's antigen presentation is at its absolute peak. You're basically pouring gas on the fire, aggressively supporting the immune system while the zombie cell is actively helping you coordinate the attack.
But eventually, that clock ticks forward. The flare gun dims, and the toxic smoke screen starts to billow out. You can't just keep boosting the immune system if the environment is turning into a swamp.
That is when you deploy step 2, a class of drugs called senomorphics. The root word there is morph, meaning to change shape or behavior. The objective isn't to kill the cell, but to alter its chemical output. Senomorphics modulate that SSP cloud. For example, they might use JAK-STAT inhibitors, like ruxolitinib. These act on internal pathways to essentially turn down the volume on the bad signals, like the IL-6 that creates the swamp, while preserving the cell's ability to present those cancer antigens.
So you are surgically silencing the smoke screen, but keeping the display cases open.
Exactly. And then for step 3, as the cell tries to build its defensive barricades, you move to a multi-axis blockade. You hit them from all sides. You combine standard immune therapies with new drugs that specifically target those defenses. You introduce molecules that rip down the CD47 don't eat me signs so macrophages can start clearing them out. And you use inhibitors that shut down the CD73 enzyme, stopping them from converting your energy into that adenosine sedative.
You dismantle their force fields while they're trying to build them. But eventually, a zombie cell outlives its usefulness. It becomes purely a liability.
And that is step 4, when you bring in the heavy artillery. Senolytics. The suffix -lysis means to cut or destroy. These are highly specialized drugs like navitoclax or ABT-263, which are BCL2 inhibitors designed to induce apoptosis, or programmed cell death, specifically in senescent cells. Once the immune system has extracted all the value from the zombie cell's alarm phase, you use senolytics to systematically eradicate the zombie cells before they can trigger those neutrophil nets or drive metastasis.
It sounds like a brilliantly orchestrated military campaign. We can call it the 4-step EMBL framework to help you remember it. Early maximize, modulate intermediate, block checkpoints, eliminate late. But I have a major logistical question. If the difference between a helpful flare gun and a toxic smokescreen is just a matter of time, how does an oncologist actually know what time it is on the zombie clock for a specific patient? You can't just guess based on how many weeks it's been since their last chemo session.
That is the most critical hurdle in translating this to the clinic, and you're totally right. Guessing is incredibly dangerous. The solution relies on longitudinal profiling. Doctors will use regular liquid biopsies, which are just specialized blood draws, to track the patient's internal clock in real time. They monitor cell-free DNA, cfDNA floating in the blood, and they track the specific levels of those SASP chemicals and those microscopic thumb drives we talked about.
So by constantly monitoring the blood, they can watch for the exact molecular moment when the profile shifts from a helpful alarm to a toxic swamp, and that biological shift dictates their next move.
It transforms the physician from someone who just administers poison into a master conductor. They are orchestrating the immune system's response, perfectly timing their interventions to trick the evolving tumor ecosystem into destroying itself.
Which brings us to a really profound realization at the end of our deep dive. We've seen how the very treatments designed to cure cancer create this highly dynamic, evolving ecosystem of zombie cells that possess the power to both cure and kill. The old model of simply trying to violently carpet bomb every last cell is being replaced by something vastly more sophisticated. I want to leave you, our listener, with a broader thought to mull over. We have spent this time focused entirely on cancer, but cellular senescence, the accumulation of these zombie cells, happens naturally in all of us as we age. They drive chronic inflammation throughout the body.
That is the exact frontier that medical science is standing on right now.
If a tumor is this highly dynamic ecosystem, and the future of medicine is acting as a master conductor, perfectly timing the modulation and removal of these zombie cells, how might this concept of cellular timing apply to the rest of human biology? Could this time-aware approach to managing the senescence clock be the key not just to outsmarting cancer, but to treating the fundamental diseases of aging itself? It's a paradigm shift that changes how we view the timeline of our own bodies. Thank you so much for joining us on this deep dive. We will see you next time.